For our lesson today, we're going to be going through the formal assessment 3. You guys did this for me last week and I would like you to mark it today, but I'll show you how to do all of that rather than just giving you the mark scheme. So, question number 1. Animal cells are eukaryotic. You should be highlighting and annotating any key terms already. Name 3 structures that are present in prokaryotic cells but absent in animal cells. Okay, so prokaryotic means bacterial cells. That should be annotated. And this question was generally answered quite well, but quite a lot of people put flagella, which is not one of the answers. You can get a mark if you said a capsule, a cell wall, a loop of DNA, plasmid, ribosomes, but you have to have said either 70S or small ribosomes. Anything in a squiggly bracket like this is essential. Pili or mesosome. The reason why flagellum would have not been accepted is because the question asks what's found in a prokaryotic but not an animal cell. A sperm cell is an animal cell but it has a flagellum, so it does not make sense to put it there. Question number 2. The cell wall of a plant cell contains cellulose molecules. Complete the table by placing a cross in the appropriate box to indicate if each statement is true or false. Now, cellulose is made of beta-glucose. That's true. Starch and glycogen are made of alpha-glucose. It's got 1 to 4 glycosidic bonds. If you remember, when you've got a molecule of glucose, we start with carbon 1, 2, 3, 4, 5, and 6. If something is joining in a straight line, then the bonds are between carbon 1 and carbon 4, which is what we see in cellulose. It's a straight chain, so yes, that makes sense. Now, if we had a bond 1 to 6, we would also have molecules of glucose branching off, which we don't actually see in cellulose, so that's false. And finally, magnesium atoms, we don't see that in cellulose either. So 4 potential marks here. The next slide was a magnification question, which actually was not as good as I expected it to be. A few people were measuring the nucleolus rather than the nucleus, and the question asked you to give it in micrometers, which again some people did not do. Now, to calculate the diameter, we know that that's the entire length of a circle. And if you remember your magnification triangle, I goes on top, I am goes on the bottom, with the top being the size of the image. This is what we can actually calculate. You would have used a ruler to find out the length of this nucleus. We need the actual size and we need magnification. Now, we have magnification, we can get image size, which means the question is asking us about the actual size, the actual diameter of this nucleus. So if you were to cover up this A, it will tell you that to solve the equation you need to take the image and divide it by the magnification, which is exactly what we have to do. Now, I have not got this printed, so I can't exactly remember what the length of this was. I believe it was about 5 centimeters, which would have been 50 millimeters, which would have been 50,000 micrometers. And all you have to do is whatever length you got, as long as it's around that, around 5, I think it was 5 or 6, divide that by the magnification to give you your answer in micrometers. Okay, now you have to convert to micrometers either before you do the division by magnification or after, but the answer has to be in micrometers. And this is what the mark scheme actually said. So the correct measurement of the diameter divided by magnification. So whatever your length of the whole nucleus was, I don't really mind here, as long as you've divided it by 6,000. And the second mark comes if you have converted it to micrometers, remembering that each millimeter is 1,000 micrometers. Then we move on to the next question. Scientists investigated the body mass index of male office workers aged 40 and the type of breakfast they ate most regularly. The BMI is calculated using this formula, and it tells you what category someone falls into in the table below. Now, this is the graph they have shown us, which we can already start to try to analyze without even looking at the question. We were told that a healthy weight is from 18.5 to 25. So, for simplicity's sake, you can always draw this as a line. This is the healthy BMI line. Interestingly, every single type of breakfast eaten by the men in this investigation resulted in them having quite a high BMI, higher than healthy. So we had some multiple choice questions to go with this. Put a cross in the box to complete each of the following statements. The graph shows that people who ate dairy products have a lower mean BMI than those who ate bread. Well, the dairy products are quite high and the bread is here, so no, that's incorrect. Fruit and vegetables have the lowest mean BMI. No, that's not true. It's cooked cereal. No breakfast have a higher mean BMI than those who eat meat and eggs. Well, meat and eggs is the second, but there is still a higher BMI there for dairy products, so that's incorrect, which means that the last one must be true. Ready to eat cereal have the highest mean BMI than those who ate cooked cereal, and that's correct. Second question, the graph shows that the mean BMI for every group sampled in the investigation indicates that they are overweight. And already, because we drew the line where healthy BMI is, we can see that this one is true. For the next one, the units for BMI. Now this was a question where mostly people said this. Now this was the equation, and by answering A, you're saying kilograms meter squared, which doesn't make sense. What you mean to say is kilograms per meter squared. Sometimes it's written with a slash, but if you're doing it in purely mathematical terms, you do it kilograms to the power of -2. Almost. It's still a squared, but it essentially means kilograms per meter squared. There's always the minus there. So the answer is actually B. And then the final question here, the scientists ensure that the data were valid by repeating the investigation with, and the answer is larger sample size. It's the same as you doing an experiment with more repeats, so with more people. You don't want to be adding new variables in, because that's confusing the investigation more. Now the question after that says, suggest how the scientist ensured the investigation was valid. And again, you need to understand what the word valid is to be able to answer this. Validity means whether you can be sure that you're measuring what you actually mean to measure. Now in this investigation, scientists were looking at the BMI and how that's affected by the breakfast that someone eats. They already told you in the question that they're making sure that they only do this for male workers and that they do it for people aged 40. Okay, so they're already trying to make it a little bit more valid, but the question is asking for any other things they should have done, because this is not enough. Male office workers aged 40 is not enough to make this a valid investigation. So, remember, anything in a squiggly brackets is essential. For example, using participants with the same or similar levels of activity or exercise, using the same or similar volume of breakfast. If you said amount or quantity, that does not get you a mark. Using the same volume of meals or drinks during the day, something other than breakfast. There's no point doing an experiment where everyone's eating porridge for breakfast, but then they eat whatever they want for the rest of the day, because you can't be sure that it was the breakfast that's changing the BMI. You want to make sure the trial duration is the same for everyone. You want to control for any other health factors. Smoking can affect BMI, general fitness, stress can affect it, so those things should be kept as similar as possible. They should all have had the same starting mass or BMI. They should all be eating the same type of breakfast for the whole investigation, and their body mass should be measured at the same time of the day. Body mass obviously differs if you measure it in the morning or measure it in the evening after you've eaten, and again, that could affect someone's BMI, so everyone should have theirs measured at the same time. Next question. Some people avoid eating breakfast in an attempt to lose weight. Using information from the graph suggests why eating no breakfast is unlikely to lead to weight loss. And again, the question says using information from the graph. That means that there are marks available for doing that. So let's refer back to the graph. Now we're looking at eating no breakfast. This is it here. If you read the BMI of people, of the mean BMI, it's 26.9. If we compare it to something like this one, fruit and vegetables, sorry, that's 26.4. So already we see eating no breakfast gives a higher mean BMI than eating fruit and veg. And the marks are available for you here. So the subjects who ate no breakfast had a higher mean BMI than those who ate, and then you can give an example of one of the other ones, like fruit and veg, ready to go cereal, cooked cereal, or bread. You can also get that mark if you said it was the third highest BMI, after dairy products and meat and eggs. Reference to a suitable calculated difference. So rather than just quoting numbers, you need to show some kind of calculation. And this could have been something as simple as saying, well, they have 26.9, this is 26.4, this minus that is a 0.5 difference. And this is what the marking point there is saying. The idea that the metabolic rate of someone who skips breakfast is influenced or is lower, and that means that they are not burning up calories as fast. Or, and some people put this, the idea that people may become hungry during the day because they've not eaten breakfast, and so actually they're ending up picking more food to eat. In the next question, cooked cereal such as porridge contain a high proportion of dietary fiber. This helps to lower blood cholesterol levels. Using the information in the graph, again, marks available for this, and your knowledge of blood cholesterol suggests why a breakfast of cooked cereal could reduce the chances of developing cardiovascular disease. And these are things like strokes, atheromas, coronary heart disease. So again, we're going to use information from the graph. We're going to look at the cooked cereal. Okay, this is the line. It's got a BMI of 25.4. Now that's still higher than a healthy weight, but it's only 0.4 higher, healthy weight being 25 or healthy BMI being 25. So out of all these options, it looks like cooked cereal is the best. The marks are available here. People who eat cooked cereals have the lowest BMI of all the groups. Again, manipulation of figures. You've done a calculation by comparing what's the BMI of someone who eats cooked breakfast versus the healthy BMI. The idea that lower BMI helps to reduce blood pressure, the idea that dietary fiber cannot be digested, the idea that dietary fiber helps to lower the absorption of cholesterol or increases the absorption of cholesterol. And finally, lower cholesterol reduces the risk of atherosclerosis. Next question was about the cell surface membrane. So the fluid mosaic model has been developed from the knowledge of the structure and properties of cell membranes. It can explain how molecules enter and leave a cell. Describe the structure of a cell membrane. You may use a labeled diagram to help support your answer. And the majority of you have chosen to draw this when I marked your original. But if you chose to purely describe it, you can only get those marks if you actually refer to exactly where the molecules are. So I'll show you the mark scheme first, and then I'll show you a drawing that you could have done. So you do get a mark if you just say that it's a phospholipid bilayer. You do get a mark if you either have drawn the phospholipid bilayer correctly, with the heads facing outwards and the tails facing inwards. And if you didn't draw it, then you can get the same mark by explaining why phospholipids are orientated the way that they are. So the fact that the heads are hydrophilic, attracted to water, or the tails are hydrophobic, repelling water. You can get a mark by showing that there are proteins in the membrane, either described or shown, and you can get an additional mark by saying or showing where they are. So extrinsic proteins being embedded in the membrane, whereas intrinsic proteins going all the way through the membrane. If you just said that there are glycoproteins and glycolipids, you don't get a mark. You have to have them described. So what are they? Where are they specifically? Alternatively, if you've drawn them, that's fine as well. And then finally, the idea of cholesterol within the membrane. Again, described where it is or shown. Those of you who have chosen to just write this as an answer would have probably gotten about 3 marks, but the other 2, because you just mentioned that there are glycoproteins and cholesterol, you wouldn't have gotten that unless you say exactly where they are. So the easier thing to do here is to draw it. So this person has drawn a phospholipid bilayer and labeled it. They've got both peripheral or extrinsic protein and intrinsic protein. They have a glycoprotein here with a carbohydrate chain. They have got cholesterol in the correct space. If you have drawn cholesterol here in the middle, you don't get a mark. If you have drawn the phospholipids really far apart from each other, you don't get a mark, if you notice it here. Not if gap between phospholipids is too large. Okay, so they should be touching. You should not have a big gap in between them, otherwise you do not get that mark. Any one of those would get you your 5 marks. The second part of that question was suggest 2 properties of molecules that enable them to enter cells by diffusion. So simple diffusion without the use of proteins, and that's of course if something is small. If you said size alone, that does not give you the mark, because I'm not sure whether you're referring to large or small molecules. Molecules that are non-polar or not charged, lipid soluble, so can dissolve in lipids, and the idea that they are recognized by specific protein receptors. The ones that were most common were the first two, small and not charged. Next bit, facilitated diffusion and active transport are two ways in which molecules are transported across cell membranes. Describe one similarity, so one thing that's the same, and one difference between facilitated diffusion and active transport. The answers for similarities could have been the use of a carrier or a channel protein. If you just said protein, you don't get a mark. You have to say carrier or channel because both of them use those. Or you could have said that it helps to transport polar or charged or hydrophilic molecules, so molecules that cannot go through diffusion alone. For a difference, the idea that active transport requires ATP, whereas facilitated diffusion does not. What I saw a lot is people saying one side of the story. So I've seen a lot of people say active transport uses ATP, but then they stop the answer there, and I cannot give you the mark unless you say, but what about facilitated diffusion? Similar here, active transport moves molecules against the concentration gradient, and you can see it here. It's moving molecules from a low concentration to a high concentration, whereas facilitated diffusion goes down the concentration gradient, so from area of high concentration to an area of low concentration. Okay, again, you need to say both sides of the story. You can't just say facilitated diffusion moves molecules from high to low concentration and completely ignore active transport. Type 1 diabetes occurs when beta cells in the pancreas do not produce insulin. Stem cells are produced from skin and can be used to replace these beta cells in mice. The skin cells can be stimulated to become pluripotent stem cells. Place a cross in the box next to the correct definition to complete the following statement. Pluripotent stem cells are. Now, if you remember, we start with totipotent stem cells, then pluripotent, and then we get multipotent stem cells. The first two answers begin with the word specialized. Now, already we know that pluripotent stem cells are not specialized. They do not have a particular function in the body other than differentiating into other cell types. So they are still undifferentiated. A lot of genes are still switched on. So we know that those two cannot be true. Next one, unspecialized cells that can differentiate to give rise to almost any type of cell in the body, excluding totipotent cells, whereas D is the same answer, except this time we're saying that it can become totipotent cells. Now, pluripotent stem cells cannot turn back into totipotent cells naturally, so C must be the correct answer. They can differentiate into any of these, but they cannot go back to make totipotent cells. Only totipotent cells can turn back into themselves and also form extra embryonic cells. Question 7 was one that was generally not answered well. Type 1 diabetes occurs when beta cells in the pancreas do not produce insulin. Same scenario as we've seen before. The pluripotent stem cells were injected into the mice. After 8 weeks, these cells had developed into insulin-secreting beta cells. So we've gone from an undifferentiated cell into one that now has a particular function, one that is differentiated. Describe how these pluripotent stem cells become specialized. And we have to be referring to what is happening to the DNA and which parts of the DNA are transcribed and translated into the protein. And this is an answer that's worth 4 marks, and I think there was maybe 2 people who got full marks on this. Everyone else got about 1 or 2. So this is definitely something that you need to revise. We start with the idea of there being some sort of stimulus, so a chemical, something that's going to cause genes to become switched on. You did not get this mark if you said genes being turned on. Those switched on genes then become transcribed, or mRNAs produced from them. Remember, anything in squiggly brackets has to be in the answer. The mRNA is then translated to produce a protein. And finally, the idea that the protein modifies the cell in some way, or the idea that the protein determines the cell function. Okay, so whatever the genes are that are switched on, that's the gene that's going to get transcribed into mRNA. That's the gene that's going to get translated into a protein. Then we move on to some genetics. Marfan syndrome is a disorder that affects elastic tissue. Mutations in the FBN1 gene can cause Marfan syndrome. The gene itself codes for a protein called fibrillin 1. Marfan syndrome is caused by a dominant allele. The diagram shows the inheritance of Marfan syndrome in one family. So this is a pedigree chart. Now remember, anything that's colored in black means that someone has got the disease. Anything in white means that the person has not got the disease. They do not show it in their phenotype. The first question is, which of the following are the genotypes, so the alleles, of individuals S and T? Okay, those are the individuals now that we're looking at. Now, we can help try to figure this out by looking at what's happening in this particular pairing here, because this pair has got a child without the disease, and because we know that Marfan syndrome is caused by a dominant allele, if you don't have it, it means that you have two recessive alleles. We know that this person has inherited the small m from this parent, the other small m from this parent. Now this parent does not have the disease, so they must have two small m's, whereas this parent does have a disease, so although they have a small m allele, they must also have the dominant allele because they do have a disease. So this parent is heterozygous, whereas this parent is homozygous recessive along with their child. Now if that gives us the genotype of this person, and the genotype of person S again will be similar to these individuals here. If they do not have the disease, then they must be recessive, homozygous recessive. And that leaves us with S being MM and T being big M, small m. So the answer is C. The next bit is the probability of child W having Marfan syndrome is, and you can do a simple Punnett square to figure it out. You've got this parent, you've got this parent, and you can see that the probability of a child having Marfan syndrome is 0.5. There's a 50% chance of offspring getting this disease. Remember, you cannot be a carrier for a dominant disorder. If you have the dominant allele, you're going to show it in your phenotype, whereas a carrier is someone who's got an allele but is not shown in their phenotype. Question 9. The diagram below shows part of a DNA molecule. Place a cross in the box next to the molecule represented by the letter X. And if we were to zoom in at this single nucleotide, you hopefully remember that we've got the circle is a phosphate ion, the pentose sugar is deoxyribose, since this is DNA, and then the last part is the nitrogenous base, and that can be adenine, thymine, guanine, or cytosine. This is a phosphate, so you get a mark if you've answered that. Place a cross in the box next to the molecule represented by the letter Y. Now we get told that this is guanine, so you have to remember what base is complementary to guanine, and that's of course cytosine. And the reason that is because they have 3 hydrogen bonds between them. So the bond between bases, the bond that actually holds the 2 strands of DNA together, is a hydrogen bond. Now you do not need to remember the exact structure of guanine and cytosine, but you do need to remember that there's 3 hydrogen bases between them, whereas adenine and thymine have 2. The next questions on genetics was actually pretty well answered. A lot of people got 4 marks. A few people got 3 because of the last marking point, which I'll show you in a minute. Achondroplasia is an inherited condition that results in restricted growth in humans. The condition is caused by a dominant allele. Fetuses which are homozygous for the allele are rarely born alive. That means big A, rarely born alive. 2 parents who have achondroplasia would like to have children. They are concerned about the risk of their child inheriting 2 dominant alleles and dying before birth. In the space below, draw a suitable genetic diagram to show the probability of a child from these parents growing up without achondroplasia. Okay, so that's what you're trying to work out. So, as always, we follow the same steps as we did in lessons. So we need to first figure out the genotypes of the parents. Now, if they were homozygous dominant, big A, they would not have survived. Those people die at birth. But because the parents do have achondroplasia, they must have the dominant allele. So both parents are big A, small a. They're both heterozygous, and they both have achondroplasia. The next step is to show the alleles, which is just separating the alleles and putting circle around them. This is showing the gametes, sorry. Then you put that together into a Punnett square. And these are the 3 marks that most people have achieved. And then the last mark, which people still forget to do sometimes, is to show which genotype links with which phenotype. A lot of people put things like achondroplasia, dies before birth, achondroplasia, or no achondroplasia, but not linked it with the actual letters. So if you've given that underneath, that's fine, or you could have drawn arrows. So here I'm showing that the AA means unlikely to be born alive. Big A, small a is achondroplasia, and 2 small a's, homozygous recessive, is healthy. Those need to be linked. And that means that the probability of having a child completely without the disease is only 25%, or 1 in 4. And that's the actual mark scheme. So we need a genotype of the parents. We've got that. We need alleles in gametes. We've got that. Possible genotypes of the children and corresponding phenotypes. So these need to be linked. And then finally the probability of 1 out of 4, or 25%, or a proportion 0.25%. This question was also relatively well answered. Draw a diagram to show molecule produced when 2 molecules of R join together during a condensation reaction. Now again, when you are annotating your exam question, you should highlight the word condensation reaction and try to figure out the definition. And a condensation reaction is the formation of a bond through the loss of water, which already gives you quite a big clue as to what you're supposed to be drawing. Now this is a molecule of glucose. You get marks if you have correctly drawn the glycosidic bond, that you have shown that water is produced, and that everything else has been drawn correctly. And I will show you 2 examples of this. Here we've got a correct glycosidic bond. Okay, so we've got a bond, oxygen bond between the carbons. So that's a mark there. This diagram has the same. That's a mark. Both of these diagrams show you that water is created, which is correct. But then the diagram on top has some inconsistencies. So here it's showing that oxygen is bound to the hydrogen, which is bound to the carbon, which is incorrect because hydrogen is only capable of forming 1 bond. So here it should be OH, but this way. And then also there's a couple of hydrogens missing on carbon 5, so 1, 2, 3, 4, 5. Yeah, carbon 5 is missing the hydrogens, which this graph does have. Okay, so this one would be 2 marks, and this one would be 3 marks, because they have shown that a glycosidic bond is correct. They have shown water leaves, and this one does have everything else correct as well. Question 11. Over 20% of the population in the UK is classified as obese. Obesity is a significant risk factor in the development of cardiovascular disease. One way of estimating if a person is obese is to find their body mass index, which with the formula we've already seen earlier on. And again, it gives you all the categories that someone may have. Calculate the BMI of a person who has a mass of 95 kilograms and a height of 1.75 meters. So the first thing you do is you square the height, and then you divide the mass by the height, and this question was answered well. The answer should be 31 or 31.02 or 31 without any decimal places. And if we try to figure out where that is on the table, 31 falls here. So obese class 1 is the answer you need to put. I did have a few people just put obese, but I can't give you the mark because there's 3 classes of obesity, so I need to know specifically which one you meant. And next question. People in this category are more likely to develop high blood pressure than people with a healthy weight. Explain why someone who has high blood pressure is at a significantly higher risk of developing cardiovascular disease. So again, atheromas, coronary heart disease, strokes. For this question, because it was a starred question, it means that the spelling of key terms must be correct for you to get that mark. Key terms are any of the things that are italicized. So you start with saying that high blood pressure causes damage to the endothelium of the artery. Notice that these are in squiggly brackets, so they have to be included. They would accept if you said lining of the artery, but if you just said wall, you do not get a mark. I saw quite a lot of people put damage the blood vessels. It's only arteries that we're worrying about because they have the highest blood pressure. So it has to be arteries, has to be endothelium, or the lining. Otherwise you don't get this mark. Now, when we get this damage, the first thing that happens is white blood cells try to rush in. We get this inflammatory response, which again, inflammatory has to be spelled correctly. Over time, the white blood cells and the cholesterol accumulate to form an atheroma. You would have also gotten that mark if you said the word plaque. And that means that because you have this narrowing of arteries, you have higher chance of developing a blood clot. You would have gotten that mark if you said the word thrombus as well. You get a mark for saying that the lumen narrows, so the space in the artery gets smaller, and that means that you're increasing blood pressure even further because you're reducing the lumen of the artery itself. Okay, so that's the idea of this positive feedback. High pressure causing even higher blood pressure when this process is done. This question was another one that was answered quite poorly, and this really limited quite a few people because it was a 6 marker. LDL cholesterol found in plasma binds to receptor proteins and is taken into cells by endocytosis. A gene found on chromosome 19 is responsible for making the receptor, and this is the receptor here. Familial hypercholesterolemia is an inherited condition. The recessive allele makes normal LDL receptor proteins. The dominant allele makes a receptor that does not function. Explain why people who do inherit a dominant allele have a risk of dying early. So what is it about the fact that they don't have this that makes them more likely to die? And of course, if you remember LDL, the low density lipoprotein is the bad type of cholesterol, and that means that the LDL remains in the blood. You have to have said blood or plasma, or that the cholesterol is not taken into the cells. If there are no receptors, the LDL does not go into the cell. The fact that the dominant allele makes a different shaped protein, a different primary structure or a different shape. Okay, so the protein may still be made, but it's the incorrect shape, and so LDL cannot bind. It could be that the cell is just making fewer of the normal LDL receptors. So again, we're not getting as many in. The fact that the cholesterol remaining in your blood plasma increases plaque formation or the formation of arteriomas and narrowing your arteries. If you've got these narrowed arteries and higher chance of a blood clot, you're not getting oxygen into your muscle cells. And if you're not getting oxygen to your muscle cells, like your heart, you're going to have a greater risk of heart disease because the cells cannot respire, or a heart attack, cardiac arrest. Question 13. A student investigated the effect of caffeine on human heart rate. 3 males of the same age were given cups of coffee containing caffeine. Their heart rates were measured 10 minutes after drinking coffee. 2 hours later, they were given cups of coffee with no caffeine, and after 10 minutes, their heart rates were measured again. The results are shown in the table. So again, heart rate in beats per minute. Remember, we saw that already with the BMI, the -1. It means per minute. The student concluded that caffeine increases human heart rate, analyzed the data to explain why these results may not support the conclusion. So what is there to say that actually no, there is no effect of caffeine on heart rate? And the first thing we can do is just look at the data. We've been given the mean heart rates with the standard deviation. Most people ignored this. They did not say anything about the standard deviation. So if we look here, the range of heart beats here, or the standard deviation, is between 70 and 78, whereas for this one is 70 to 72. And those overlap. So that means actually there doesn't seem to be a difference between drinking coffee with caffeine and drinking coffee with no caffeine. We also see that male number 3 did not have a change in their heart rate at all. This was the mark that a lot of people got. As we said earlier, standard deviations overlap, so the difference may be due to chance and may not be significant. And finally, we do not know what concentration of caffeine we use, so we can't really be sure whether it was enough to cause an effect, or you could have said that the sample size was too small. They're only using 3 males, so we cannot conclude that caffeine increases human heart rate. They have not used enough participants in this experiment. Next one, describe how this investigation could be improved. Now, a few people had some good ideas here. Take into account the resting heart rate. All we got was their heart rate after drinking caffeine and not before. We need to know what their heart rate was previously, because maybe it was quite similar to when they drank caffeine. We could include females in the samples to make it a more representative study. And we could have included a range of ages in the study as well. We've only used males of the same age. Take into account any previous coffee drinking habits. So have they already drunk coffee earlier that day? Do they drink coffee quite often? And finally, because heart rate can be affected by exercise, that needs to be taken into account as well. So that means that some of the participants might be quite active, the others might not be, and that again could be influencing the heart rate and not the caffeine itself. Question 14. Graph A shows a correlation between systolic blood pressure and a percentage of stroke patients having a second stroke within a few months. The ischemic stroke is the type of stroke where there is a blockage in the artery, and that means blood is not flowing through and there is no oxygen, and so brain tissue dies, and that results in a stroke. And so we've got some points here along with some error bars, and generally we can see that the higher your blood pressure is, the more likely someone is to have a second stroke after having one already. The second graph we get is showing us blood pressure again, but how it differs depending on whether you have a high salt diet or a normal salt diet. And the third graph shows us the effect of additional risk factors on the number of deaths due to cardiovascular disease. So the more risk factors you have, so things like being male, being older, high stress levels, smoking, high salt diet, eating a diet that's high in cholesterol, that makes you more likely to die of cardiovascular disease. Men who smoke have an increased risk of having ischemic stroke. Analyze the data to discuss what advice could be given to a smoker who has had one ischemic stroke already to reduce the risk of having another stroke. Now, because the command term here is analyze, you have to be showing some kind of calculation here. You should not just be giving general trends. I saw a lot of people saying things like there's a positive correlation between pressure and the percentage of patients having a second stroke. While that's correct, that's not quite analysis, that's just describing a graph. So you need to always show some kind of calculation. Now, you could get marks for any of these. The fact that the advice should be to reduce blood pressure. To get a second mark, you need to show some analysis of data. That means you need to show some kind of calculation. Maybe what is the difference between having blood pressure of 130 and blood pressure of 190? You could calculate the difference there. Another thing could be to discuss the fact that they could be having antihypertensive drugs to try to reduce blood pressure or to reduce smoking. They should also be reducing salt in their diet, but that should be supported by the analysis of data. So if we're looking at this graph, you can't just say high salt diet gives you a higher blood pressure. You have to calculate a difference. So you could choose these 2 points and calculate what is the difference here. Reduce blood cholesterol levels is another piece of advice you can give. Reduce saturated fat intake, use anticoagulants, reduce BMI, increase exercise. All of these are pieces of advice you can give, but because this is a 6 marker, it's actually a leveled response. So if you just gave 5 different bits of advice but have not referred to the graphs at all, you will not get full marks. So this is quite useful to use here. To get between 1 or 2 marks, you need to have had a discussion of how to reduce one risk factor with reference to one graph. So you could have said something like eat less salt, reduce salt intake, because I don't know, high salt intake can increase your chances of having a stroke by 40% or whatever the calculation is that you did. Or discuss how you could reduce 2 risk factors if you have not referred to graphs at all. And this is where a lot of people were stuck. So they gave some advice but have not referred to the graphs in enough detail, so I could not give them the next level, which is between 3 to 4. If you discussed how to reduce 2 risk factors and reference that to 2 graphs, you can get 3 to 4 marks. And if you show good analysis of data as well. And finally, to get level 3, so between 5 and 6 marks, you need to show how to reduce 3 risk factors with reference to at least 2 of the graphs. And to get that, you should have shown sustained application of relevant evidence of analysis, interpretation, and evaluation of pieces of information. So to get this level and to get level 2, you should be criticizing some of these. Maybe they're not the best graphs. You know, we can see here that the error bars overlap. Here we can see that we've got a high salt diet and a normal salt diet. We don't actually know what they mean. They've not given specific quantities of salt here, so you can criticize that. You can say that the study did not go for long enough. All of these are relevant and can give you the marks you need for this question. For question 15, this is the last question now. Prostate cancer is one of the most common forms of cancer in adult men. A number of investigations have been carried out to study prostate cancer in a large number of men in Denmark, Finland, and Sweden. In a third investigation, prostate cancer in identical and non-identical male twins was studied. Data were collected on the probability of one male twin developed prostate cancer if his twin brother had prostate cancer as well. Explain why identical twins were used in this investigation. The answers for this were quite good, actually. Loads of people were saying that identical twins have the same alleles or the same genotypes, or that they are genetically identical. If you said they have the same genes or the same DNA, you will not have gotten this mark. You have to be quite specific here. It's the same alleles, not just the same genes and the same DNA. Finally, for this 2 marker, the differences in identical twins must be due to environmental factors. So we're using them to try to see whether prostate cancer is caused by an environmental factor or whether it's caused by a genetic factor. And this is what the final graph is. So we've got the probability of both twins developing prostate cancer and their age of diagnosis, age at diagnosis. The straight line is the identical twins, and then dashed line is the non-identical twins, which you always want to color them in to just help you figure things out. Analyze the data from this investigation to evaluate the likely causes of prostate cancer. So by the end of this question, you should form an idea of what it is that causes prostate cancer using the data. We need to be referring to the data. And so, from the graph, we can see that identical twins do have a higher probability of both twins getting prostate cancer. And that means that there are genetic effects. The probability is higher in identical twins than non-identical twins, which suggests that there is a genetic factor involved. Now, because the question asks you to analyze, you should be quoting some numbers here. So you may want to choose any point, any age, and just show a difference by reading off the graph. It could be that it means that both of the twins have a mutation in their DNA, and that's why they both get a cancer.